Okay, guys, let's take a closer look at this pyruvate dehydrogenase reaction, the overall process. So remember, again, we're up here at the beginning of this reaction with pyruvate. We're going to run this oxidative decarboxylation reaction to form this hydroxyethyl TPP, right? Now, this reaction up to this point, as I mentioned before, is essentially identical to one you saw before when we looked at pyruvate decarboxylase in the first step of alcoholic fermentation by yeast, right? So if you'll recall, this cofactor, TPP, thiamine pyrophosphate, is this really interesting molecule, this pyrimidine-like ring attached to this thiazolium ring. And then, of course, a couple of phosphate groups over here. But the business end of this molecule, if you remember, is that methine carbon right there and that proton, right? Because if you recall, that methine proton is unusually acidic compared to a normal methine proton, right? By virtue of this sort of imine, like quaternary amine in this thiazolium ring, there is a fantastically good sink there that allows that proton to be abstracted. And it's thought in the active site of enzymes like this, there's a little acid-base catalysis going on that pulls the proton from one of the aromatic amine groups on the other side of the ring that then facilitates the exocyclic amine on the other side acting as a base to abstract that methane proton from the thiazolium ring to create that resonance stabilized carbanion. Remember, this is a really unusual situation where that carbanion is unusually accessible because of the acidity of that proton and the structure of that thiazolium ring. And if you recall, this is an awesome nucleophile, right? Carbanions are one of the best nucleophiles you've got. And if you bring that pyruvate carbonyl down close by, boy, will this baby happily attack that electrophilic carbon of that carbonyl group. So when that happens, right, of course, again, remember that this is also this this deprotonated version of TPP, that resonance stabilized carbanion, sometimes also known as an ylide. When it attacks to form that so-called addition compound, remember the name of the game here is we want to decarboxylate this alpha keto acid. And one could, in principle, as I've mentioned before, well, we could try and drive this thing down by having these electrons from this carboxylate group come down here and split this guy here. But if all you had was that carbonyl, well, that's not going to work really good, right? But if you do a little covalent catalysis here by forming this this TPP adduct, you have over here on the nitrogen of that thiazolium ring, that positively charged imine like quaternary amine. That's a really great place to stick some electrons. That's a nice, deep, energetically stabilizing hole. So now when you peel off the electrons from that carboxylate group to split that bond, you now have somewhere for those electrons to go, right? To really stabilize that carbanion that would be essentially impossible to create with just the pyruvate, that alpha keto acid alone, right? So we drive the CO2 off, right, to now form this sort of resonant stabilized carbanion of this hydroxyethyl TPP. The hydroxyethyl TPP, this guy can also take up a proton to form a hydrated form of hydroxyethyl TPP. And we sort of completed the first step of the reaction. Like I said, this ought to look familiar to you from the pyruvate decarboxylase reaction, because in pyruvate decarboxylase, we would take this addition product right here, do a little acid-base chemistry on this sucker, and kick off acid aldehyde. But we're not going to do that. Pyruvate dehydrogenase isn't going to do that, just sort of like stop there. We're now going to use the sort of free energy available in this compound to synthesize that activated thioester, right, which is the next step of the reaction. Now, all of the stuff I just talked about, that all, remember, happened up here on the E1, right, that outer shell of the pyruvate dehydrogenase complex. And so we've created that hydroxyethyl TPP in the active site of that E1. Now, the next important thing that happens is that lipoic acid residue is going to become really key, right? And I haven't shown you what that is yet, but that cofactor is covalently attached to a side chain of a lysine residue, excuse me, on the transacetylase. Remember that E2 that's right down in the core of the complex, right? And this is what that dihydrolipoamide residue looks like. First, we've got a lysine side chain on a ketamine, right? There's that epsilon amino group, and we form this amide bond with lipoic acid, which is this sort of five carbon chain with a carboxylate hanging off one side, form an amide bond. There on the other end of this molecule is this little haptan, like disulfur containing five-membered ring that happily and readily forms a disulfide bond. Should be kind of reminiscent of Cleland's reagent, if you guys remember what DTT looks like. So really quite readily forms a disulfide bond. So what we're going to do is use this as an electrophilic center to drive that synthesis of a thioester bond by using that stabilized carbanion on that hydroxyethyl TPP, right? And this happens again with on the E2 enzyme, right? So here's our hydroxyethyl TPP. In this form, it's now ionized. There's our activated resonance stabilized carbanion. That's going to attack that one of those electrophilic sulfurs in that disulfide bond to basically thioesterify that carbonyl carbon of that acetate group, and it's going to migrate then from the TPP over to one of the sulfurs on that lipoamide group. In this form, it's dihydrolipoamide here. It's hydroxyethyl lipoamide now, once we've made that thioester, right? Notice something else. This reaction also results in, so if you imagine, right, this carbon, that carbanion is going to attack that sulfur. These guys are going to come up here. They're going to take that proton here, come back down, right? We regenerate the carbanion on the TPP. You'll notice also that sulfur group has been reduced, right? Which means this guy has taken up electrons, right? Turns out in both cases, right? So we've simultaneously generated a thioester and reduced that disulfide bond. So this dihydrolipoamide is both an acetyl carrier and an electron carrier, right? Okay. So next thing we need to do is make some acetyl-CoA. That's the second half of this, the E2 reaction, the trans acetylation reaction. So here again is our acetyl lipoamide, all right, group. And we're going to bring in some reduced coenzyme A, and we're basically just going to transfer that thioester bond. It's a trans acetylation, right, reaction. Energetically neutral. The sulfur is just going to attack that carbonyl, facilitated by positioning the active site to then generate our acetyl-CoA, which is the activated 2-carbon carrier product we are after. But notice, leaving behind reduced dihydrolipoamide, right? Right. Whereas lipoamide was what we had before. Now we have dihydrolipoamide. We've reduced it. Okay. If the reaction stopped here, we'd have made acetyl-CoA, but we only get one reaction out of this. Because remember, to get the formation of this acetyl lipoamide to go, we need to have a reasonable electrophilic center for that carbanion on the hydroxyethyl TPP to attack. Right? That requires that disulfide bond to be there. Well, now we're reduced, which means to finish this reaction out and regenerate our enzyme, regenerate our catalyst, we've got to re-oxidize those two sulfurs to a disulfide bond. That's where the E3 comes into play. All right. So we're now going to take that reduced lipoamide, that dihydrolipoamide, and some FAD. And we're going to run a 2-electron, 2-proton transfer, as we know FAD can do, to form FADH2 and regenerate our lipoamide disulfide bond. Interestingly enough, this enzyme doesn't stop here. It now transfers those electrons from FADH2 to NAD+ to generate NADH, which is released, and regenerate the FAD for another round of the cycle. Okay, that's the basic 4 steps, right? So we have 1, 2, 3, and 4 steps of the overall pyruvate dehydrogenase reaction. Now, the weird thing, if you're thinking about this for a second, is like, okay, we have 3 enzymes, but that complex was huge, remember? I mean, this thing is gigantic. It's twice the size of a ribosome. And the key thing, if you're paying attention, was that E2 with the lipoamide moiety on it, down there in the core, But the dehydrogenase that gets it started, it's all up here on the outside. How does it even get there? How does that even work? Okay, well, the answer is that that lipoamide group, right? If you remember, it's kind of on a long little chain, but that's even stuck on this, the lipoamide domain of the transacetylase is on this big, long swing arm. So each one of these trimers, each one of them has this long extended domain, right? That's on this so extended loops. And then out on the very tip of that domain is that lysine residue that has that lipoamide group attached to that amide bond, right? And there's that reduced disulfide. And essentially what happens, this enzyme, this acts like a little robotic, a little, like, I guess a chemical robot, right? Where this is like a little robotic arm that's gonna move back and forth between these different active sites as it makes acetyl-CoA. So to see what that looks like, let's sort of go through the sequence of how this is thought to work. Here we are at the beginning of the reaction, ground state of the enzyme. Here's the core of our enzyme. There's the E2 down in the core. Here's the E3, the dihydrolipoamide dehydrogenase, and there's the pyruvate dehydrogenase with some bound TPP in it, ready to go. So this enzyme binds some pyruvate, decarboxylates it, as we saw, to generate this hydroxyethyl TPP in the active site of the pyruvate dehydrogenase enzyme. And only at the point where that happens, only at the point where this hydroxyethyl TPP is formed, does something happen. There's a conformational change in the E1 subunit that has successfully formed hydroxyethyl TPP, and it opens a channel. It opens this 20+ long pore channel, like 20 angstrom long hydrophobic channel that is perfect for that extended chain of that lysine residue and that dihydrolipoamide and maybe a bit of the protein to then insert down into the active site where that dihydrolipoamide has just been made. And it positions that oxidized lipoamide group near the hydroxyethyl TPP so that esterification can take place to form the acetyl lipoamide. When that acetyl lipoamide is made, the swing arm then gets ejected out of the E1, right? There's again a subsequent conformational change sensed in a way I don't think this is understood, but the acetyl lipoamide moiety, once it's formed, gets ejected from the E1 pyruvate dehydrogenase subunit, and it's thought to then fold over to a neighboring E2 subunit where the transacetylation reaction takes place. When this is loaded, right, acetyl lipoamide group is loaded, this enzyme will now bind some reduced CoA to form, then through the transacetylation reaction, acetyl-CoA. Now, when acetyl-CoA has been made, and the lipoic group, the lipoamide group is now dihydrolipoamide, it's reduced lipoamide, the swing arm is ejected again, and it moves now to the E3 enzyme, the dihydrolipoamide dehydrogenase, where there's a waiting FAD that reoxidizes the dihydrolipoamide to lipoamide, where it is the point then ejected again. The dehydrogenase enzyme then binds some oxidized NAD, which re-reduces the FAD and moves the electrons off to the NAD to form NADH, excuse me, right? So you can imagine this sort of fascinating way in which this must be happening down in the core of this enzyme. This little core of all these, 8 trimers, each with its own little swing arm, zipping around back and forth, right, on sort of, the sub-second, probably millisecond, time scale, furiously zipping about, buzzing around like little knitting needles, making acetyl-CoA, getting re-reduced, going back and getting some more dihydrolipoamide, going back and getting some more, another acetyl group, and then just swinging around like that. It's sort of a fascinating image. And it's a major theme in enzymology. Like I think I mentioned before, people used to think that all this biochemistry used to happen with these isolated enzymes floating around, and the substrates would just diffuse around. And there's a certainly a certain amount of that going on in biochemistry, but that was biased by the way in which biochemists for decades isolated and purified these enzymes. And so they thought about these reactions working this way. And what's becoming true as we've learned more and beginning to appreciate the structure of organized living systems like cells. People are recognizing more and more that these enzymatic functions are organized in many cases in very specific geometric fashion in super complexes. And this is one really dramatic example of how multiple different enzymes that are in a particular pathway are put together so that the sort of channeling of intermediates happens in a very efficient way between different active sites. In this case, it's not even diffusive channeling, right? Because we've got this like, covalently attached swing arm that's moving back and forth. So this is like a really extreme example, but there are lots of growing examples of situations where even a product from one reaction has to diffuse to another enzyme. Those guys are frequently being found in larger super complexes, right? Okay, so one last thought before we end this lecture. Why go to all this trouble of making a thioester, right? I mean, why not just do this with an ester, right? It's a pain to go through doing all this. Well, remember, right, as you probably remember, acetyl-CoA is an activated carrier, right, of that acetyl group, right? If you just made an ester, you just esterified that carbon, right? You basically are blowing energy, right? Because that ester is lower in energy than a thioester by virtue of the fact that an ester has a resonance structure that a thioester can't populate, right? So the free energy available relative to sort of like the hydrolysis products of an ester versus a thioester is significantly lower. So in essence, if you tried to do something like this with a thioester, you would have significantly less free energy to work with in the subsequent step that we then want to use for synthesizing ATP, which is the ultimate direction we want to go. So the thioester essentially preserves some of that free energy available in that acetyl group, or the acetyl group we created when we decarboxylated pyruvate. We preserve some of that free energy that we can then use in subsequent steps, right? Okay, see you next time.